Hi friends, good morning. How are you? Hope all are fine. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about the structure of spleen. The spleen is the largest lymphoid organ in our body. It is an intraperitoneal organ, that means all of its surface except the hilum is covered by a layer of visceral peritoneum. Underneath the peritoneum there is a capsule that covers the spleen. The capsule consists of fibroelastic connective tissue. This diagram explains the internal structure of the spleen, and this is the capsule. The capsule covers the spleen and it consists of dense irregular fibroelastic connective tissue. This connective tissue contains contractile cells known as myofibroblasts. The contractile activity of these cells help the spleen to discharge stored blood into the circulation whenever necessary. In addition to this, the capsule also allows the spleen to increase its size whenever necessary. The capsule gives rise to a number of septa. These septa are known as trabeculae. These trabeculae penetrate into the parenchyma of the spleen and partly divide the tissue. Next is, like other organ, spleen also consists of stroma and parenchyma. The stroma consists of a network of reticular connective tissue that provides support for blood cells and also for cells of the immune system like lymphocytes, macrophages and dendritic cell. Next is parenchyma. The parenchyma of the spleen is divided into two morphologically and functionally distinct compartments known as red pulp and white pulp. Red pulp and white pulp. The red pulp and white pulp, they are the two compartments of the parenchyma. They are separated by marginal zone. The red pulp and white pulp are separated by a marginal zone. This is the marginal zone which separate the red pulp and the white pulp. First of all, we will discuss about red pulp. The red pulp, it consists of splenic cord and vascular sinusoids. The splenic cord is also known as cord of Billroth. The splenic cords are cellular aggregations that are supported by reticular connective tissue, and the splenic cord contain different types of cells like blood cells, macrophages and plasmocytes. Other is vascular sinusoids. The vascular sinusoids are blood-filled space, and the splenic cord appear as stripes. The blood flow slowly through the vascular sinusoids. These are the vascular sinusoids. The blood flows slowly through the vascular sinusoids where they become exposed to macrophages from the splenic cords. So the red pulp acts as filter for removing the toxin from the blood before it enters into the systemic circulation. White pulp. The white pulp of spleen consists of lymphoid follicle, marginal zone and periarterial lymphoid sheath. The periarterial lymphoid sheath consists of central artery which is surrounded by a sheath of lymphoid tissue. The lymphoid tissue is distinguished into two layers, The inner layer contains only T lymphocytes, whereas the outer layer contains both B lymphocyte and T lymphocytes. The branches of the central artery, these are the branches of the central artery, are surrounded by lymphoid follicles. These are the lymphoid follicles. Two types of lymphoid follicles are seen in the white pulp. They are primary lymphoid follicle and secondary lymphoid follicle. The primary lymphoid follicle contain mainly small immature lymphocytes. When the primary lymphoid follicle mature and increase in size, it is known as secondary lymphoid follicle. The secondary lymphoid follicle differ from primary follicle in having a germinal center. The germinal center is the site where the B lymphocytes acquire the ability to produce antibodies. These are the three components of the white pulp. One is lymphoid follicle, another is marginal zone, next is periarterial lymphoid sheath. Follicle means lymphoid follicles. Two types of lymphoid follicles are seen. Primary lymphoid follicle and secondary lymphoid follicle. Then there is a marginal zone. This marginal zone is between red pulp and white pulp. And next is periarterial lymphoid sheath, which consists of a central artery that is surrounded by a sheath of lymphoid tissue. So this is known as periarterial lymphoid sheath. The lymph is a highly vascular organ. Blood from the splenic artery enters into the spleen through the hilum. From there, the splenic artery divide and form many branches which enter into parenchyma following the course of trabeculae. These artery branches throughout the parenchyma and gradually decrease in diameter. In parenchyma, these arteries are surrounded by periarterial lymphoid sheath, and the artery which is present in the central region is known as central artery. The central artery divide and form many branches which enter into the marginal zone. From there, the arteries further move into the red pulp region where they transform into arterial capillaries. From arterial capillaries, blood flow into the splenic cord. In the splenic cord, old and damaged erythrocytes are destroyed by the macrophages, and from splenic cord, blood flow into the vascular sinusoids. Due to the lack of junction between the endothelial cells, only the normal and healthy erythrocytes become able to enter into the vascular sinusoids, whereas malformed and old damaged erythrocytes as well as antigens like microorganisms and cellular debris are destroyed by the macrophages which are present in the splenic cord. From vascular sinusoids, blood flow into the trabecular vein, and from trabecular vein, blood ultimately exits the spleen through the splenic vein. Blood flow freely in the vascular sinusoids. This type of circulation is known as open circulation, and open circulation is the characteristic of the human spleen. This is about the structure of spleen. Thank you friends. Please share, like and subscribe this video. Also please subscribe my YouTube channel known as Some Online Class. Thank you very much.